Welcome back, everyone, to Einstein's Eyes. I am Carl Rosen, oculoplastics, neuro-ophthalmology in Anchorage, Alaska. I've been here for 30 years, trained with Jon Ditkoff.
Hi, I'm John. I'm a general ophthalmologist specializing in cornea. I've been in New Jersey now for over 30 years. I met Carl back at Albert Einstein where we did our residency, hence Einstein's Eyes. So welcome to our channel.
We had a pretty good response to a talk about Neuralink, Elon Musk's company, and brain chips that interface and potentially can offer vision to a non-seeing person as long as they have an intact visual cortex or brain substance that allows for vision to be interpreted. Today, we're going to expand on that and talk about the world's first whole eye transplant. Now, this stuff sounds completely crazy, but it happened, and it happened at NYU Langone, so New York University School of Medicine, with Dr. Eduardo Rodriguez as the team leader. It was a massive team. It was over a 20-hour operation on a former veteran who had an electrical injury. Aaron James, actually 46-year-old, who suffered electric injury that destroyed his eye and part of his face. So it was a partial face transplant and eye transplant. The interesting thing is that most people think about eye transplants as what we as ophthalmologists know are really cornea transplants. Here's a cornea that's been transplanted. These stitches are in the cornea, and you take a donor cornea from a cadaveric eye, and you replace the damaged or diseased cornea in a living person. And John, you can expand on this.
Yeah, so I specialize in cornea. This is a very nice corneal transplant. So what was done was a full thickness cornea was removed from a donor eye from someone that passed away. Usually it's a suitable person, like a younger person, sad to say, and they have a very healthy cornea. And then the host cornea is cut, as you can see at the edge, right where the stitches are. There's that white little kind of faint line. That's where the cornea was cut and removed. And then the new cornea was placed in its place and stitched in.
Most people think of as a transplant. It's really not a full eye transplant. It's a corneal transplant, which is just a part of the eye. It's the front part of the eye, the sort of the dome-shaped window that allows light into the eye. Transplant a whole eye is a completely different matter. And you must also transplant or resuture the muscles. There's 6 extraocular muscles: the superior rectus, inferior rectus, lateral rectus, medial rectus, superior oblique, and inferior oblique. Here's a cross-section of an orbit. You can see the eyeball. Here's the cornea in front. Here's the optic nerve. This is the internal carotid artery. This is the blood supply to the optic nerve and to the eye. Now, you can see the muscles here. Now, there's 3 muscles that this diagram is showing, but there's actually 6 that would need to be replaced or put back on the eye to make the eye move properly. And if you were to transplant an eye, you would take a piece of the optic nerve and somehow suture this nerve to the remaining stump so that roadway or the conduit for the visual system to transmit vision into the brain. You would also have to reattach the ophthalmic artery and the ophthalmic vein so that blood could come in and feed the tissue and blood could go out and take away the deoxygenated blood, bring it back to the heart for further oxygenation. The optic nerve carries about 1 to 1.2 million axons that come from something called the retinal ganglion cells. And these are critical cells that allow for vision. And let's look at the layers of the retina. There are 12 or more layers of the retina. The top part is the inside of the eye, and the bottom part here is the sclera. So you're coming a cross-section from inside the eye down into the substance, the white part of the eye, or the sclera that we're looking at when we look at an external picture of an eye. Now, the nerve fiber layer consists of the axons that go into the optic nerve, and then this layer right here is called the retinal ganglion cell layer, and this is super critical to transmit vision to the eye. Now, these other structures are important too, something called the internuclear layer, the outer plexiform layer, the outer nuclear layer, the pigmented epithelium, and then the receptor layer consists of the rods and cones that are responsible for night vision and color vision. This shows you the complexity of the retina that allows for vision. November 2023, a 21-hour surgery on Aaron James. As I mentioned, a military veteran, a 46-year-old electrical worker, was injured. He survived. Eduardo Rodriguez was a team leader at NYU Langone. These surgeons and the rest of his team transplanted part of his face and the entire left eye. I mean, this is miraculous. They had a 12-millimeter stump of donor optic nerve that was preserved and bathed in a stem cell slurry, and the retina was also perfused on the table. Now, let's fast forward. Now, that in and of itself is a miracle, and there's just miracles happening all the time. And Neuralink is a miracle, and this is a miracle. So we're going to fast forward 1 year later, and what happened? Well, the eye is still alive. There's normal pressure in the eye. There's a healthy retinal blood flow. There's light-evoked signals on MRI imaging, but there's no conscious vision yet. This is the hard part. So why didn't the vision return? Well, the retinal ganglion cells that leave the eye and travel through the optic nerve synapse in something called the lateral geniculate nucleus, which is inside the brain. And then from there, they go into something called the visual cortex. Those connections have not happened. A barrier that gets created, there's scar tissue that's created that we have to get beyond. And the retinal ganglion cells themselves enter something called apoptosis, which causes damage to those cells after about 2 weeks if they're not reconnected. There's 6 elements to a toolbox for optic nerve regeneration that Jon and I are gonna talk about and highlight today. And we're not going to go deeply into it, but we're going to highlight these, and they're super interesting. One is called gene editing, and that has to do with CRISPR. Now, CRISPR is called the clustered, regularly interspaced short palindromic repeats. You don't need to know that. That's technology that has been recently discovered and utilized, particularly for sickle cell disease, where you can edit out genes that are not good and put new ones in. And these are essentially gene editing technology that lets scientists cut and modify DNA segments with precision. Gene editing allows for growth mode of these retinal ganglion cells and the axons, and primarily experiments have been done in mice, and their axons have reached into the brain. Another category is called bioscaffolds, and there's something called CNTF, which is ciliary neurotrophic factor. This is a protein that promotes survival and growth of neurons used in regenerative gels. Third is 3D conduits, and these are engineered lattice tunnels that guide axons while nutrients flow through them. Now, these have been tested in rabbits without inflammation, so another promising area. The fourth is stem cell retinal ganglion cell sheets. These are skin cells that have been reprogrammed into brand new ganglion cells. Work has been shown dendrites actually growing in monkey eyes. A fifth, optogenetic relays. Some axons grow and they make the surviving cells light sensitive. Early trials have been attempted in something called inherited retinal dystrophies. And these are a group of genetic diseases called retinitis pigmentosa, where the photoreceptors are slowly destroyed. Optogenetic relays are being trialed for this particular category, inherited retinal dystrophy. And the trials are running now. The final one I want to talk about, the final category, is electrical coaching, where you have transorbital alternating current stimulation, boosted field sensitivity in optic neuropathy patients. Putting it all together, you can think of 3 phases. You have the hardware, which is the eyeball, you transplant the eye. You think of the firmware, like the gene therapy plus the scaffolding that pushes the axons across a gap. It could be a glial cell gap or a scar gap. And then the software, and that's a Neuralink style chip on the lateral geniculate body or the neuron that records signals, machine learning algorithm techniques to get a fresh visual map. Now, you can liken this to the way cochlear implants have been used to teach the brain to hear again. There's ethics, there's cost, there's equity involved, there's donor eligibility. Currently, only about 3% of deceased donors or cadaveric eyes are really useful. The current surgery and rehabilitation costs are probably $1 to $2 million. And then the patient has to be under lifelong immunosuppression. Now you have to question, what does that mean for quality of life? The trade-offs, the gains, subjective, and really an individual has to make those decisions. 3 questions. Can any blind person get an eye transplant? Only if the rest of the eye is gone, but the brain pathways are intact. And this is a small subset. What is the timeline for partial sight recovery? Right now, people think 5 to 10 years for some light perception and 15 years or more for real images to be identified. What about retinal chips? You've heard about brain chip interfaces like Neuralink is doing in the occipital cortex, which is what blindsight got FDA approval for. Retinal chips are great for photoreceptor diseases, like I mentioned, retinitis pigmentosa, but are not good, or they're useless for optic nerve injuries or if the optic nerve is severed. There you have it, folks. A really interesting topic. We're going to touch on this just like we're going to stay on top of Neuralink. But John, what do you think? Fantastic stuff, John, huh?
Fantastic, overwhelming. I'd like to see it in my lifetime, like real. I mean, I think the exciting part, of course, is the way I've always told my patients is when the retina is damaged and the nerve is damaged, it's not that different than being paralyzed. So it's the same research and same excitement that are going to regenerate people's ability to walk again. I think that's not my expertise. Neither is this, but that's beyond the scope of this. But similar,
Right.
yeah.
Right. And there are stem cell experiments. There are neuroprotective and neuroregenerative chemicals that are also being used that we didn't touch on. And I also wanted to mention that the Advanced Research Projects Agency for Health, which is a governmental agency, is funding optic nerve regeneration and eye transplants. And that is what NYU Langone is part of. There is a consortium that is being funded by the government. I think this is fantastic. So we look forward to this. We look forward to reporting back to you and discussing the advances in this really wild technology. But folks, the future is bright and things are looking good. And we'll be here to talk about it. So thank you. Please subscribe to the channel. We are growing slowly, but please subscribe for John and I to bring you these. We love it. And if you like it, please hit that like button, and we will see you on the next one.